Very good morning, dear students. So we have been discussing unit number 3, proteins and amino acids and their characteristic features. Then the next topic specified in your syllabus, ionic charge of protein molecule and electrophoresis. First, briefly we will discuss the ionic charges of protein, and we will discuss electrophoresis. So it is a technique to study the molecular weight of unknown proteins. And ionic charge of protein, you have already seen that in the previous class. A peptide molecule comprises of amino acids, and the amino acid has 4 important parts. One is an amino group, next one is a carboxylic acid group, and next one is the side chain R, and next one is the hydrogen atom which is attached to the alpha carbon, and that is known as alpha hydrogen. And whenever appropriate conditions, so there will be a deprotonation will take place from this carboxylic acid group, and that will go and bind to that proton is bind to the NH2 end, and there will be protonation will take place. As a result, this will be a zwitter ion. Okay, and in one end there will be a positive charge, and another end there is a negative charge, and there are high level of ionization is happening. And the ionic charge of protein is at a high pH value. The net charge of most of the protein is negative, where they bind to positively charged matrix in anionic exchanges. If the protein is a negative charge, they of course they will migrate to the positively charged matrix. When the environment is at the pH value is equal to protein's Pi, we will discuss that. The net charge is 0, and the protein is not bound to any exchange, and that can be eluted out. Okay, when the environment is the pH is equal to that of the Pi of protein, okay, then the net charge will be 0, and in that condition the protein will not go and bind to any exchanges, thus the protein can be segregated. And this Pi is known as isoelectric point. That is exactly that the term we are using, pH or I or IEP. Okay, isoelectric point. So isoelectric point is nothing but the pH at which a molecule carries no net electrical charge or electrically neutral in the statistical mean. That is known as a isoelectric point. Okay, isoelectric point, the pH at which the molecule carries no net electrical charge or the electrical charge is neutral. Okay, that is known as isoelectric point. And the net charge on the molecule is affected by the pH of the surrounding environment and can be more positively or negatively charged due to the gain or loss of electrons, gain or loss of protons respectively. Okay, so the net charge of a molecule is affected by pH of its surrounding medium, and it can be more positive or negative charge, and due to the gain or loss of a proton. Okay, and this is the basics of ionic charge. So the protein will be having an ability to ionize in its charged form. Okay, at various pH, some pH the isoelectric point, the pH at which the molecule carries no net charge, okay, then that can be eluted out. Okay, that is known as the isoelectric point. And then another important technique in this unit we supposed to study is gel electrophoresis. So gel electrophoresis, as the name indicate, here we use a matrix with a certain gels, okay, so then polymers, okay, and electrophoresis is the process of separation of the charged molecules, okay, in a electrically charged matrix in the presence of an cathode and anode. And electrophoresis is generally used to characterize one of the most basic properties, okay, molecular mass and the charge of the polypeptide and the amino acids. And electrophoresis, gel electrophoresis can be used to determine the purity of the protein and the heterogeneity and extent of degradation in the protein and the composition of protein. So these are the major aim of performing electrophoresis, to determine the purity of protein, then to determine the heterogeneity and the extent of degradation of a protein, to determine the composition of the major subunit present in the protein. And what is the principle? And simple, you know that the cathode and anode principle. The principle of electrophoresis is the migration properties of charged species within an electric field. Okay, the migration of the charged species within an electric field is the main principle. It is simply the behavior of opposite charges attracting each other. Okay, there is a principle. The opposite charges attract each other, same charges repel each other. Okay, an electric field is established across the electrode of the power supply, and the charged ion moves in this particular electric field. Okay, there are two types of electrode, you know that, anode and cathode. So anodes are nothing but positively charged electrode which attract the anions or negatively charged component. Okay, then cathodes are nothing but negatively charged electrode which attract cation or a positively charged component. Okay, the cathode and anode terms are consistent with their redox reaction, that is oxidation reduction reaction definition. So reduction occurs always at cathode and oxidation occurs always at a anode. And cathode reduction takes place, in anode oxidation takes place. Okay, one is removal of hydrogen, next one is addition of hydrogen. Okay, so that is the addition of oxygen. Okay, that is the important concept here. So here you can see we are having a power supply, and this is the anode, positively charged electrode, that will be always being oxidized, as I said, and here the electrons will be going, okay, and here the electrons will be losing. Okay, then the cathode, negatively charged electrodes, okay, and this is being reduced. Okay, so here the movement of ion across the electric field, they are carriers of the charge. This is the normal principle. Okay, and the major factors that influence the rate of electrophoresis migration, as I said, whenever we subject it to a particular component in an electric field, what will happen? The charge will migrate. Okay, and the electrophoresis migration RF is mainly depending on three factors. Okay, the strength of electric field E, which is directly proportional to the migration rate. Second one, the charge on the ionic species, that is Q, that is directly proportional to the migration rate. Next one is the frictional coefficient of the support matrix. Okay, that is the gel, that is F, that is inversely proportional to the migration rate. So we are having an equation RF directly proportional to QE by F, where RF is the migration of the migration rate, then Q is the charge, okay, E is the electric field, and F is the frictional coefficient. And these factors can be varied in the following way. the strength of field is a function of the voltage and the power. Okay, the electric field strength is the function of the voltage of the power. Okay, and the voltage is thus proportional to the resistance across the electrode. Okay, the oxidation is proportional to the resistance across the electrode. Okay, when the current comes to play, but in short way, it is difficult to achieve high voltage across the electrode if the resistance is low. Okay, if the resistance is low, even if the current comes to the electrode in a short point of time, a short manner, it is very difficult to achieve a high voltage across the electrode. Okay, and the resistance of the solution is inversely proportional to the ionic strength or concentration of ion. Okay, when high salt concentration, the resistance is low, it is difficult to achieve high voltage and the migration rate obviously will decrease. Okay, so if the salt concentration is high, then the resistance is low, then it will be very difficult to achieve high voltage, so the migration rate will be low. Okay, the charge on an ionic molecule is a function of the isoelectric point of the molecule and the pH of the solution. I told you here the principle is based on the charge and molecular mass, then the function of Pi, isoelectric point, and the pH, hydrogen ion concentration. If Pi is greater than pH, if isoelectric point is greater than pH, then the molecule is cationic, which migrate towards the cathode. Then if the Pi is less than that of the pH, then the molecule is anionic, which migrate towards anode. Okay, if the Pi is equal to pH, then the molecule is neutral, no migration in the electric field will takes place. Okay, so thus we can alter the migration by altering the pH of the solution. So this is the condition. Thus we can alter the migration by altering the pH. The frictional coefficient of the matrix can potentially be altered. Because of this one, the frictional coefficient, one of the important parameter to support the electrophoresis migration rate. Okay, that is frictional coefficient. Then the matrix is, then we are talking about the matrix. Okay, the matrix is a gel. Okay, what is a gel? It is a polymer network or a gel, and the frictional coefficient can be increased by increasing the concentration of the gel. Okay, so the frictional coefficient can be increased, and if you increase the concentration of gel or the polymer. And the gel commonly used in electrophoresis of protein exclusively called polyacrylamide. Okay, so what is polyacrylamide? Okay, so this is the gel matrix we are using for the gel electrophoresis, for the separation of the protein. Okay, so the polymer is a polyacrylamide. So what is a polyacrylamide? It is a polymer composed of two covalently linked components. Two covalently linked components such as acrylamide and bisacrylamide. Okay, so acrylamide and bisacrylamides are the polymers. I have not shown the structure due to it is a little bit complex. Okay, so this bisacrylamide is essentially a cross-linking component of the acrylamide polymer. So in order to frame a polymer, a gel, it requires a cross-linking component, that is bisacrylamide. Then the concentration, the typical value for the acrylamide and bisacrylamide ratio is 19:1, and the total acrylamide concentration in the gel affect the migration of the protein to the matrix. Okay, so the concentration of the acrylamide gel is very, very important. The gel, if it is the gel, it will be adversely increase the the concentration of the gel and the migration of the protein. Okay, so if the concentration, if the high molecular mass proteins are separated using low concentration of the gel, okay, if the concentration of the gel is low, then high mass polymer can be segregated. Okay, then the other way around, the low molecular weight mass proteins are separated using high concentration of the polymer. So when you are applying high concentration of the polymer, the pore size, okay, will be very less. Only low molecular weight protein can be segregated. So when the molecular weight is low, the concentration is low, there will be a lot of space between the gel. The pore size will be high. Then you will be having there is a possibility of passing the high molecular weight component. Okay. Then the different proteins have a different charge for a given value of pH. That you all know. The pH will be having different proteins have different charge for a given value of pH. The protein will migrate as a function of both their mass and charge. Okay, the mass is big ones move slowly. Okay, the mass means big ones moves very slowly. The mass is very high, they moves very slowly. The mass is very low, then they moves very fast, and their overall charge. If there were some way to cause each protein to have an ideal charge to mass ratio, is there any method we can make an ideal to charge to mass ratio? Then it is possible to separate the mixture of protein based on the charge to mass effect. So that is the principle here. Okay, so if there any way we are able to make this charge to mass ratio somewhat identical, then it is possible to separate the mixture of proteins. Okay, then one of the important thing is known as sodium dodecyl sulfide polyacrylamide gel electrophoresis. One of the most important types of gel electrophoresis using for based on the above said principle is known as sodium dodecyl sulfide polyacrylamide gel electrophoresis, or simply call it as SDS page. Okay, simply call it as SDS page. So sodium dodecyl sulfate, that is known as SDS, or sodium lauryl sulfate, is an anionic detergent. Okay, why we are using this anionic detergent? That is a question. So if SDS bind via hydrophobic interaction to the protein in a stereochemistry approximately proportional to the size of the protein. Okay, so a small protein will bind few molecules and large protein will bind a lot of SDS. So due to the charged nature of SDS molecule, the protein will have an approximate constant charge to mass ratio. I told you, any method, if you are able to make an ideal charge to mass ratio, then it is possible to separate the protein from the mixture. That is what we are using by adding the SDS. Okay, when you are adding the SDS, the protein will have an approximate constant charge to mass ratio due to the charge provided by SDS, and will migrate through the gel at a rate proportional to the molecular mass. Okay, so I told you it is an anionic detergent which provides charge to the protein, and that provides that produce a constant charge to mass ratio. Due to this, the migration will take place through the gel at a rate proportional to their molecular mass. The proteins migrate towards the anode due to the charge on the SDS is negative. Okay, the charge on the SDS is negative, thus the protein will migrate to anode at under all pH condition except the acidic. In acidic condition, the process will be revert back. Okay, then the protein gels are usually performed under denaturing condition. So first, when you are performing this process, the sample preparation, first you prepare the protein sample, and you need to add SDS, and you need to heat the a protein sample in presence of SDS. Okay, so what is the reason? To fully unfold the protein and permit the binding of the protein. You know that the protein, the native form, that will be fully folded protein. We will discuss that, and when you are adding that, there will be degradation will take place and the SDS will bind throughout the length of the polypeptide. So once the SDS is bound, the characteristic isoelectric point value of the protein is no longer relevant. Okay, the isoelectric point will be no longer relevant because the SDS has already given a charge. There is no equal neutral charge here. Thus there is no point of considering this isoelectric point. Then the protein takes a negative charge and each protein will be essentially the same charge to mass ratio also. Okay, the negative, due to this negative charge, the protein has acquired the same charge to mass ratio. So this is the structure of sodium dodecyl sulfate or SDS. It has got a CH3, 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 CH3 like that. O, S, double bond O, double bond O. Here you can see S is the sulfur is the central atom. Here you can say sodium is bound to this particular region. The O has negative and positive charge. There is a, that is why it is known as sodium dodecyl sulfate. This is known as dodecyl sulfate, and it has two parts: the hydrophobic region that doesn't have any charge, and here it is a hydrophilic region. Okay, here you can see a negative charge. Okay, it's a polar region. This particular region will bind to the proteins. Okay, then so the migration, so what will happen? The migration rate, protein mass and the percentage of acrylamide gel is very important components in the acrylamide, polyacrylamide gel electrophoresis. Okay, so the greater the percentage of acrylamide in the gel, greater the frictional coefficient and slower the migration rate. Okay, if the percentage of acrylamide will be very high, then what will happen? The molecule will be only the small, greater the frictional coefficient will be high, then the molecule can migrate in a very smaller level. If the protein to be separated are a high molecular mass, and if the percentage of gel is high, the protein may not even enter the gel. Okay, so the protein to be separated are the concentration is the molecular, that is high concentration, the high molecular mass, and if the gel is also high, the protein may not even enter the gel. So it is essential to match the percentage gel to the mass of the protein being separated. So what is the percentage of the gel and what is the mass? There should be a proper agreement. So this is the concentration of acrylamide in the gel, and what are the ranges of separation of polypeptide, the length of amino acid. If it is 8%, it can be separated by 25 to 200 kilodalton molecular weight size. Okay, that means to say approximated 225 to 1800. That is what I told you. If the concentration is very lesser, what we can do, the pore size will be very less. The pore size will be very high, there will be very loose, then larger protein can be separated. If it is 10% of acrylamide, 15 to 100 kilodalton, then the concentration is increases, size is decreases, so 135 to 900 amino acid in size that can be segregated. If the concentration is 12.5%, 10 to 70 kilodalton protein can be segregated, approximately 90 to 630 amino acid can be segregated. If the concentration is 15, if it is increases, 6 to 660 kilodalton protein, 55 to 550 amino acid. Then if it is 20, then the separation of the polysaccharide range can be, the size is reduced, then 4 to 40 kilodalton, approximately 30 to 360 amino acid. Okay, that's what I told you. The concentration of acrylamide gel is very less, then it can help to separate high molecular weight protein. Then if the concentration is very high, then it will help to provide a low molecular weight protein. So when you are adding the concentration, the pore size will be decreases, so it will not allow the larger protein to migrate through the gel. So that is the reason. And again, the migration rate, protein mass and the percentage of acrylamide gel, the setting up the SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis, it is essential to know when to stop the experiment. When you will stop the experiment. This is somewhat difficult to determine since protein do not absorb the spectrum. We cannot simply see the gel to determine whether the protein have been separated or not. So the protein will be moving through the gel, okay, but we cannot simply see whether it is moving or not. Thus, it is common to use a protein sample, a small anionic dye molecule, okay, along with it, in order to see the gel is colorless. The sample whatever we are adding is also colorless, okay. In order to see the protein, the movement of the protein, you are adding along with the gel, you are adding a small light colored blue dye that is known as bromophenol blue, okay. The size of the dye molecule is selected to be very small so that there is essentially no frictional coefficient with the gel, okay. Generally the size of the dye which will be very small because it is selected to be very small so that there is no frictional coefficient within the gel, okay. The dye is chosen to be anionic so that it will migrate in the same direction as the protein and SDS complex towards the anode, okay. That is very, very important. The selection of the dye, it should be anionic also because SDS is anionic and the protein also, they are going towards anode, okay. Since the dye is anionic and small, it will migrate fastest of any component in the mixture to be separated, okay. So the dye is also anionic, so it can be migrate very fast, okay. So the dye molecule is also selected to absorb in the visible spectrum, be visually detectable while the gel is running. We can see while the gel is running where the gel is reached, whether the separation, where the molecule from the top to bottom, where it is reached. In the protein or SDS or dye mixture, there will be having the protein, SDS and dye mixture is loaded on the top of the gel, that is cathode, and when the dye molecule, that is known as dye front, okay, the front region of the dye, the traveling region, okay, reaches to the bottom of the gel where the anode is there, the power is turned off and the experiment is halted, okay. When it is reached to the top to bottom, once the blue colored moves and we can towards down and reaches to the cathode, from cathode to anode, we can stop the experiment, we can turn back the plug-in, okay. So although the gel support provides some friction to molecular motion, as soon as the power is turned off, the separated protein will begin to diffuse. So once the protein is turned off, what will happen? Through the gel, there is a possibility to diffuse, okay. So there are freely insoluble in aqueous solution, because there is a solution, aqueous solution, there are freely insoluble. So in order to prevent this, immediately the gel is treated with an acetic acid and methanol solution, which causes almost all protein to precipitate, okay. So when you are adding acetic acid and methanol, this will cause the protein to precipitate. There will not be any diffusing of the protein will take place, and this process is known as fixing the gel. Now the separated protein will not diffuse, okay. So that is the important thing. Then the fixed proteins are still invisible but must be visualized by the staining, and we cannot see, we can see only a blue colored regions there, okay. And the fixed proteins are not clearly visible, and because everywhere it is light blue color, okay. So you need to stain the gel by staining. So what is the common stain we are using to stain the SDS page? That is known as Kumasi Brilliant Blue, okay. A component called R250, Kumasi Brilliant Blue R250, okay. So the fixed gel is incubated. Once the process is over, one day you need to incubate the gel, you need to take it and you need to add Kumasi Brilliant Blue in a tray, and you need to incubate with the Kumasi Brilliant Blue, and then the stain. After one day you need to wash the stain of the gel and incubate in a weak solution of acetic acid and methanol. Again you need to wash because the stain is over accumulated there, that you need to wash it off. So the stain will not bind to the acrylamide and will wash out. However, it remains strongly bound to the protein in the gel, okay, and this takes in the deeper blue color. So you can see a deeper blue colored regions once you see the proteins, okay. And then once the SDS treatment, the protein will migrate as a function of their molecular mass, okay. The approximate molecular mass of the separated protein is the function of their migration distance. So it's very, very important. The approximate molecular mass of the separated protein is the function of their migration distance. If the series of protein with a non-molecular mass is analyzed in an identical gel under identical condition, then there is a standard curve can be established that can be used to determine the molecular mass of the solution. And when you are using this one, certain standard or reference you need to use, okay. So here the molecular weight, okay, molecular weight standards are follows. Commonly used standards are phosphorylase B, they are protein. The molecular mass is non-molecular mass, that is 94 kilodalton. So the bovine serum albumin, that is 64 kilodalton. So ovalbumin, that is 43 kilodalton. And carbonic anhydrase, that is 30 kilodalton. So soybean trypsin inhibitor, 20.1 kilodalton. So alpha lactalbumin, 14.4 kilodalton. So various controls or reference molecular weights standards are available. You can use all those things, okay. Then this is what an example I have told here, an example of a molecular mass standard and two unknown sample of the analyzed on 10% of SDS and stained with the Kumasi brilliant blue. So this is the standard mass, okay. Here I told you 92 kilodalton, 68, 43, 31, 20.1 and 14. All those things you can see, okay. So here these bands are different bands after staining with Kumasi brilliant blue. This is the unknown sample very near to the line 13 kilodalton. By comparing with, you can also find out the mass. And this is the sample. There is a sampling wells are there. Here only it is loaded. This is unknown sample migrated towards this direction. This is known as dying front. I told you the entire blue color will be where it is reached. Okay, that is known as dying front. From here, the cathode to anode it will migrate, and here you can see the bands. Okay, these are the proteins. So now this will be this you can stain with the Kumasi brilliant blue. Okay, Kumasi blue, and this you can compare with the size, and you can study the molecular weight. And how will you quantify the protein by using SDS page is, so the measure you can quantify the protein, you need to just measure the migration distance of the dying front. Okay, from here to here just to measure the dying migration distance of the dying front, and measure the migration distance of the protein as well. Okay, here this is the migration distance from here to here, this is the migration distance, and from here to here is the migration distance of the protein. Then what you need to do, divide the protein migration distance by the dye front distance to get a relative mobility value, that is always less than 1. Okay, so if you divide this distance by this one, okay, so that is divide the protein migration distance, so this distance from the total distance traveled by the dye, okay, that if you divide, you will be getting the relative mobility value. And there is a plot we can make, the plot of the relative mobility value. You can plot along x-axis, plot the relative mobility value along x-axis versus the log of the molecular mass along y-axis. Okay, then the relationship between the relative mobility and the log of the molecular mass should be a linear function. You can see a linear function providing a standard curve against which the molecular mass of unknown protein can be determined from the relative mobility. Okay, this is what I have told here. Okay, so the calculation of a migration distance, relative mobility, and the relationship to the log of the molecular mass yield the following. Here in x-axis the log molecular mass in kilodalton, here you can see, and in y-axis and x-axis the relative mobility. Okay, and if unknown, here the whatever the protein sample, here you can see it is migrated the unknown protein sample. Okay, so this is the unknown protein sample, this is protein sample 2. Okay, so here you can see this is the unknown protein sample. Here it is 26.0 kilodalton. When you are comparing with this one, approximately 26.0 kilodalton by comparing with this one. Okay, but above 20.1 kilodalton. Okay, then here you can see the second one is the 21.0 kilodalton, 4 kilodalton. Okay, so here you can see this is 20, this is just 21.1 kilodalton. Okay, so like that you can compare. So the unknown, that is sample 1, has a value of 1.43 for the log of the molecular mass or mass of approximately 26.9 kd. So second one will be the value of 1.33 for the log of the molecular mass or the mass of approximately 21.4d. Okay, here you can see this, you can make the the relative mobility versus log molecular weight, and you visually checking the unknown versus the standard in the above gel indicate the below gel indicate the calculation of the calculation appears to be correct. Okay, so this is, don't worry about this thing too much, this is just an example. Okay, you don't need to bother about how you will get to all these values and all these plots. Okay, just to understand that by plotting this linear function you will be able to find the molecular weight if the sample has in your sample has multiple component with multiple molecular weight component by comparing with the mass, standard mass. Okay, so that is the important thing. Then this is all about SDS page, and what are the variation of SDS page. Now SDS page, there are a lot of variation you can see. Okay, one is known as discontinuous SDS page. Here that is a composite of one gel on the top of the other. Okay, one gel on the top of the other to achieving the focusing of the protein bands, okay, which are sharper band yielding a greater sensitivity and resolution. Second one is gradient gel electrophoresis. Okay, that is, instead of pouring a single percentage of polyacrylamide gel, you need to pour a gradient with high concentration at the bottom and the lower concentration at the top. This permits the resolution of a wide range of molecular mass samples. Next one is silver staining. Generally we stained with the Kumasi blue, as I said, and you will be getting the light blue color after the process. Okay, so but this Kumasi blue is not able to detect if the concentration is very low level of proteins. So then you need to use silver staining. It is a more sensitive type of stain in comparison to Kumasi blue, which allows the detection of very lower concentration of the protein. Even if very less amount of protein is getting precipitated, separated, if you add silver stain, you will be getting a good result. Okay, so you will be able to get. Then next one is, or transfer the resolved protein bands. Okay, from here you need to cut the gel and you need to transfer this particular gel to or band to this transfer the band to a secondary support such as nitrocellulose. Just cut the band and the gel you stick it with a nitrocellulose and probe with other reagent like fluorescent labeled antibodies or antibodies. Okay, that is another thing that easily can be studied. Then the page with no SDS. Okay, so instead of adding, because when you are adding SDS, the protein will denature, you know that actually. Okay, so then what will happen? The SDS will disrupt the complex. Okay, so but if you wanted to study such a kind of a protein without disruption, then you need to use the page, polyacrylamide gel electrophoresis without SDS of native proteins if you wanted to study, to allow the detection of non-covalent complex of 2 or more protein. I told you this SDS will disrupt the protein chain. Okay, so the SDS page with the addition of reducing agents such as beta mercaptanol or dithiothiol. Okay, so etc. This reagent will reduce the disulfide bond and separate the polypeptide chains that are connected by such bonds. Okay, so that is another important variation. So there are a lot of advancement you can make to perform this SDS page. Okay, now I will show you the entire, whatever I have told you might have not understood properly. Then I thought I will show you this particular picture. Okay, so this is what is the protein. Okay, this is a one subunit folded form of protein with a certain intrinsic charge. Then another protein, it has got one chain here. This is another protein which has got two chain. This is this chain A, this is chain B and chain C. We wanted to segregate different chains, 3 chains. Okay, so what you need to do, all the sample that will be placed into a small tube, a pen of tube or microfuge tubes. And as I said, first step is you need to add SDS, sodium dodecyl sulfate, and you need to heat. So what will happen? SDS will bind the linear, it will make the chain into linear, it will bind throughout the this one, and it will make the chain negative charge. Okay, then the denatured protein with a uniform negative charge. Protein will be denatured, it will be negative throughout, the negative uniformity will be there. Then this particular sample, using a micropipette, you need to load this thing into the gel. Before that, you need to prepare 2 glass plates. You should have there are separate electrophoresis apparatus. This is thick glass plate. There you can, a tray you can prepare, and required percentage of acrylamide and bisacrylamide you can mix and you can make. There are 2 types of gel. One is known as a stacking gel, in which the place you wanted to add the gel. And how will you prepare this particular wells? By adding something called as comb. Okay, comb. Okay, so this comb will be placed, and you add the first you add the in a small tray you add the stacking gel. Stacking gel, the concentration of acrylamide and bisacrylamide will be different. Okay, so here you are adding the sample. Okay, then what will happen? You need to place a comb. Okay, a comb like this. Then after solidification, you remove it. Then you will be having this particular shape. Okay, so this place you can add the sample. Individual port you can add one sample. And second one is known as a resolving gel or separating gel. Okay, one is stacking gel. You need to add the sample. I told you, when you are mixing with the dye, it will migrate in the blue color, and it will travel from top to bottom. It will travel. Then the glass place, you will keep it here, and this will place in a buffer solution. Okay, buffer solution. Okay, then you will add this particular sample in this wells, number 1, number 2, number 3, along with the control. Okay, what is control? Standard non-molecular weight proteins. Okay, as I said, bovine serum albumin. Okay, all such kind of things, soybean trypsin inhibitor, all those things. Okay, then what will happen? You will be able to place another chamber, that is cathode chamber. This is here. This particular region should be separated. Okay, you should be added the buffer solution here. Here also buffer solution. The cathode is touched with the buffer, anode is also touched with the buffer, and here you will be having a plug-in, and this that is cathode, this negative charge, and this you are having another plug-in, anode, that is negative charge, and this will be connected to the power supply. When current passes, what will happen? The protein start migrating depending upon the migrating distance. So here you will be having the your control non-standard side. Here you will be having the protein with all those 3 chain. This is sample 1. Here you will be having sample 1. This is sample 2. Here you will be having sample 2, sample 3. Larger proteins will be form the band very near to the well. Smaller proteins will migrate very fast, and they will travel fast. Okay, so this you will be having, and you can stained with the Kumasi brilliant blue, and you will be getting like this actually, or silver stained. Silver stained you will be having, it will not look light blue color. If you stain with silver stain, you will be having the dark color. Okay, like this you will be able to. Here you can see, once this is done, this can be identified. This is the gel. So this is the overall process of electrophoresis, especially sodium dodecyl sulfate polyacrylamide gel electrophoresis or SDS page. So that's all, dear students. So we can conclude, we have discussed in this particular class the ionic charge of proteins. We have discussed what is ionic charge, okay, when it will get an ionic charge, what is isoelectric point, what is gel electrophoresis, what is the principle of electrophoresis, what is anode and what is cathode, okay, what are the factors which influence the migration rate RF, then the gel matrix, what are the major components that is used for gel matrix, for the preparation of just gel. Okay, then what do you mean by sodium dodecyl sulfate? Okay, as DS. In this polyacrylamide gel electrophoresis, what is the migration rate, protein mass, and percentage of acrylamide gel is used for this process? And how will you visualize the separated protein? How will you determine the molecular mass? And how will you quantify the molecular mass? First to determine, you need to use standard. Quantification, you need to plot a graphs. Okay. Then later you have to calculate the result. How will you calculate the result? And what are the several variations, advancement in SDS page? Method. Okay. These are the all about protein purifications by SDS page and their ionic charge, dear students. These are the textbook. If you look principles of biochemistry by Leninger, whatever I have told, you will be having, you will be getting entire things. Okay, you can with a simple diagram you will be able to see. Okay, in addition to Leninger textbook, you can refer all those textbooks as well. Okay, so thank you, dear students, for watching this particular class. If you are having any clarification, you can write it to me, or you can post below the video. Thank you very much.